So welcome everyone to one of the last sessions of this fantastic Human Cell Atlas meeting. My name is Sarah Crome, myself and Jesse Gillis, who will be coming up in just a little bit. Really want to lead a very interactive discussion. So we're going to start this off today with kind of some examples of typical ways as a wet lab scientist, if we find something in our single cell data set, different approaches that at least I've tried to apply within my own team to validate these functions. But really where we want to go with this is more talking about big picture, talking about how we can move beyond these kind of basic assays and basic kind of validations that we're currently doing, and get to big picture questions like how we think about cell function in general, and what are new approaches that we could use to validate these predicted cell functions that we're finding. Just by way of example, I'm just going to use a recent publication where we were looking at the role of, we're looking at healthy human kidney. So in this particular case, we were looking at living kidney donors. The organs were used for transplantation, and the goal was really, we wanted to use this as a map to compare any aberrations that could occur, be it with deceased donor transplant, be it with any type of autoimmune disease, or any kind of transplant related adverse outcome. And so we just kind of started with mapping this healthy human kidney, and we had 9 individuals where we took the entire biopsy. So this is an 18-gauge needle biopsy. And then we had about 10 that we had to immune enrich, and that was because at least in the healthy kidney, we were unable to find a very large number of immune cells, and that was one of the main things we wanted to focus on. And so this really reflected what we'd see if we look by histology, that they're about less than 1% normally when we look by histo. And so to zoom in on the immune cells, one of the first things that came up is when we were able to look at the immune cells, what approaches do we want to use to validate the cell functions we were predicting of these resident immune cells we were finding in kidney. So one example is validation through other data sets. If we find a novel population in our particular tissue source that we think could be contributing to homeostasis, do we see it in other kinds of data sets? And so obviously here there's issues with data set modalities, the types of tissue sources that all have to be considered. And so an example of where this came up is we found this alternatively activated myeloid population that was a very dominant myeloid population. And we wanted to see if this was telling us something unique. And the reason for this is at the same time, the Eppelman lab was putting out some papers where they were really describing this fetal derived macrophage population that was present in both mouse and humans and could be tracked by certain markers. And that included FOL2, LIVE1, for example, but they really struggled at the time to find these myeloid cells within healthy human kidney data sets that were available, but they weren't, the prior data sets really hadn't used living donor samples. And so what we did here is to see if, okay, maybe there's something about these fetally derived myeloid cells that makes them particularly sensitive to different kinds of inflammation. If that's the case, we might see them in living donor kidney, but maybe not others. And certainly by using this one cell at a time integration method, we were able to see that they seem to be captured here. But once again, if we think they're sensitive to ischemia and these different kinds of effects, how do we actually validate that that could be true, right? We're really limited with the ability to take this beyond a simple observation and move to validating that that's in fact the case and that's what's happening in kidney. Another challenge we kind of came up with is how do we validate predicted cell-cell interactions and cell functions? So obviously, there was a lot of excellent talks yesterday on spatial, and this is very good for receptor ligand interactions, showing proximity, showing gene expression programs related to interactions. But what about if the interaction that you want to follow up on involves a secreted factor? And this may or may not even be produced within the organ you're looking at, for example, right? So in this case, we found these granzyme K T cells, which were highlighted in this morning's plenary session. We're very interested that we're finding them in kidney. What are they doing? Is there any association with age? But we really can't do much besides validating that, yes, in fact, if we look at protein level, we do see these granzyme K positive T cells that are there, and they're not co-expressing granzyme B or other preference. It's very basic, right? So how do we understand what these T cells are doing in kidney and how they could impact diseases in the kidney, especially with aging? Another example of this is when we looked at NK cells. One of the interesting findings we found was that there was amphiregulin being produced by these resident NK cells. And this is a tissue reparative factor. You normally wouldn't think of an NK cells producing this tissue reparative factor, but we can see it present within the kidney. We don't see it in NK cells in the blood. We see it decreased if we look at tumor unaffected nephrectomy samples. So it seems to be unique to this living donor kidney, but really beyond showing that there are certain cells that are expressing the receptor for this, we're really stuck with how whether we can validate whether this amphiregulin from the NK cells is kind of promoting repair within the tissue itself. And so, just thinking about big picture, a lot of the time we look within our tissue or organ, but we also need to consider what's going on within the entire human being and how this could impact the functions that we're looking at. The last kind of big example kind of show from what we've been doing is when we're trying to use machine learning methods to identify differences in sexes. So we were fairly well powered. We had 19 kidney donors. We had approximately equal representation between male and females, and we wanted to get some idea of whether there was anything we could find within our data set that would explain some of the differences in incidence of different kinds of kidney diseases. And so we decided to use machine learning approaches, as many people do, but they each have their own biases. And so to get around this, one of the things we tried to do is use multiple machine learning methods. So for example, Verimax, we also use MAST1, which takes into account type 1 errors. And we took these three, we came up with, okay, what are the predicted molecules that are expressed using all three machine learning methods after removing X and Y linked genes, or in one of the two. And obviously we can go and validate, hey, this is up or down in other kidney data sets. But that seems quite basic, right? So if we look at predicted differences in cell functions, for example, one of the big hits that came up is it looked like there was differences in the respiration capacity of male and female proximal tubule cells. So we could go and perform seahorse assays where we show if we take these proximal tubule cells from male or females, there's a difference in metabolism that we can observe. But really, if our hypothesis is this could underlay some kinds of kidney diseases, we're stuck. We're basically at a point where we can make an observation, it's supportive, but how do we prove causality? And so these are kind of various approaches people are trying to use. We're looking at spatial. Most of the time we're trying to capture just a snapshot, or we are capturing just a snapshot. So more and more groups are trying to look at various time points, looking at different organs all at the same time to better understand the whole individual that we're looking at. But we still have the issue that if we look at, homeostasis versus disease, this is a healthy individual, and this is an individual usually with an end-stage organ disease or a type of cancer that's fairly advanced. So we're not really capturing what's happening in the middle and progression. And we know from a lot of studies that each individual kind of has their own disease progression. And so these are kind of challenges, especially when we start to think about validating these communities or networks of cells. And so this is kind of something that I think is something that we really need to think about more now that we're looking at these communities of cells that are changing in health and disease. But if we're staying within the human system, it's really hard to recapitulate these networks of cells within 3D space. Humanized mouse models all have their limitations. If we move to conventional mouse models, sometimes they don't accurately mirror the genetic diversity, the impact of environmental factors, et cetera, et cetera. And as an immunologist, a lot of the molecules I look at, say on an NK cell, are not present on mouse NK cells. So oftentimes there's not a good model that I can study my NK cells function. And so how do we think about this and get over these obstacles? We're making profound discoveries, but how do we move forward to actually validating these functions in individuals? And so with that, I'm going to hand it over to my co-chair that's going to move from a very kind of myopic, these are the challenges we had, to kind of more big picture questions.
Thanks, Sarah, and thanks everyone for being here. So so I think this is an interesting topic. It involves a lot of words that I'm not precisely sure what they mean. So function, I don't really know what function is all the time, cell function particularly. Cell type wasn't in the title, but it could have been. I don't really know what cell types are. Maybe networks and validate. These are all words that we use a lot, but I'm not 100% sure what we mean by them. The point of this conversation today is partially to try to spell out what those words mean. So you can do so in two ways. One is extensionally. So you list a bunch of things we all talk about cell types all the time, so we must mean something. So we can all talk about cell types together for a while and then decide, oh, that's what we're all saying when we talk about cell types or function. So people use the words a lot. And so if you list a bunch of things, examples, that's an extensional definition, or you can really decide on what it actually means by trying to say, you know, not a prime minister is this set of people who are prime minister, but prime minister is the leader of Canada, you know, in this way, or the leader of the party or whatever. So, so I do like to think sort of broadly about these questions, I think, because I don't understand them. And so one of the ones that that I thought was informative about function, and was alluded to a couple of times during the meeting, people referred a couple of times to how this was, you know, the next great atlas after kind of the human genome as an atlas and genome function annotation in the human genome. And for those who were around 10 years ago, may recall that discussions about what functions there were in the human genome was extraordinarily controversial. So ENCODE had finished, you know, the post-pilot project was the first phase, and then it finished its kind of major phase about end of 2012, and they claimed that 80% of the genome was functional. That was the claim that made, you know, the front page of newspapers, and there was a lot of pushback about that, and these kind of arguments or discussions, it sometimes involved less typically placid language than you expect in science, are really quite instructive, I think, to return to talk about what people mean by function sometimes. So just to refer to the kind of the three main papers, which I've only listed in order in which they appeared, so nothing else. One was a very strongly worded article by Grauer et al., pushing back against the ENCODE definition of function. So saying that biochemical activity, for example, or mere existence is not a good evidence of function. And then there was a really interesting article in PNAS, which I encourage everyone to read if they haven't already, by Ford Doolittle, which isn't explicitly about function, but really goes through and tries to define function kind of from first principles. What do we mean when we talk about function? And then there was a response to it from basically the entire ENCODE consortium, trying to acknowledge the truth of some of the pushback, and coming up with this consensus image that we can mean lots of different things by function, that there's this very broad sense of function in which most of the genome is functional. And then there are these more and more restrictive meanings of function, which intersect with other definitions, such as genetic evidence and evolutionary evidence. So the starting point of the Ford article, which I'm not going to quote, but does say that all biologists probably agree that selected effect is how we're supposed to define function. So we're supposed to believe in evolution. We're supposed to believe nothing in biology makes sense except in the light of evolution, and that the way we define function is by how things are selected. And so if it's not a selected effect, it's not functional, but we can't literally observe that. And so a lot of the time we're stuck with evidence for selected effect or not. So ENCODE defined function very, very broadly. So a discrete genome segment that encodes a defined product or displays a reproducible biochemical signature, for example, protein binding or specific chromatin structure. And so that also sounds a lot like the way the gene ontology defines function. So the gene ontology is kind of a mix. There are different evidence codes, as was alluded to earlier. And also evolution is kind of implicitly incorporated quite strongly in the gene ontology by the fact that a lot of the annotations are made via orthology. So a lot of the annotations come under the assumption of evolutionary conservation or plausible evolutionary conservation, but the actual defined structure itself is not really evolutionary, despite the original purpose being about cross-species comparisons in some ways. And cross-species comparison is very challenging using the gene ontology. It's been the source of a lot of discussion on its own. So Doolittle wrote this, again, I think very lucid and thoughtful article about how we think about function, and came up with these three broad kind of categories. One is evolutionary, so evidence for function, I should say, not how we think about it, but evidence for degree of function. So one is evolutionary conservation, which is the strongest evidence according to that article. The other is experimental ablation, so which you can think of, sometimes they call it but-for causation in like philosophy, I think. So the idea that if it weren't for this factor, this event wouldn't have happened. So obviously lots of things are involved in anything happening. If I removed all the air from this room, we couldn't breathe, but we don't usually think about the fact that there's air in the room as a source of causation. So experimental ablation is usually in the context of everything else occurring. If I ablated this one feature, would you still get the effect? And there are lots of cool systems to do experimental ablations. One of the reasons I like plants is you can do experimental ablations that don't kill the organism. And you can see real phenotypic effects sometimes by ablating cell types, which in humans you wouldn't be able to do, or even in mammals. But also it makes us think about what's the appropriate model to do an experimental ablation is that will give us some sort of functional information in a cellular context. In a genomic context, maybe it's a bit easier if we know we're ablating a segment of the genome or some downstream product. And then the final definition, so the weakest, the one we're all supposed to, I would say from this viewpoint, supposed to think is so weak, it's barely worth mentioning, is mere existence. And I don't want to be too provocative, but I will say that is where single cell data is like 99% right now. We have existence, we have patterns in gene expression, and those are inferred to be functional to some degree, to some degree. And so we're mostly still at mere existence. And the excitement of going further in validation is really pushing into particular systems that allow either ablation, and also obviously there's a lot of excitement these days, as being mentioned at Lane's talk yesterday, in cross-species comparisons, which do get at some degree of conservation. So how to translate these earlier concepts from, say, ENCODE and either other domains into cell type discussions of function and validation beyond replicability. So mostly when I'm trying to validate a cell type, I just say it replicates across data sets. And I consider that pretty interesting and difficult sometimes. You know, there are batch effects, there are all sorts of tissue contexts, et cetera. You heard there were 5,000 cell types in the brain the other day. So there's a lot of challenges associated with all this complexity. So mere existence is actually quite significant across data sets and across laboratories and across systems. But there's also getting at causation and function, I think. And so to that end, Sarah and I put together these 3 questions with the help of ChatGPT. On my end, so I cheated a little bit. So I wrote the questions and then I had ChatGPT brainstorm on how to phrase them better. So the first one is a very broad question. And so these are supposed to be open. We're hoping everyone will chime in. We're not giving talks, significantly. So is what do we mean by function? So, and one question also is degree of function versus type of function. So like the gene ontology, so there's a lot of complaints about the gene ontology often, even while everyone uses it, but it does capture lots of types of function. Whereas conservation only really captures degree of function. So when we say something is deeply conserved, we have degree of function. When we say something is in this particular gene ontology category, it's type of function. And then as Gary mentioned, I think it was yesterday, or it might have been day one, there's multi-scale. There's the idea that things could have multiple functions at different levels. So I guess as an open question, is everyone convinced about evolution as the right way to think about function, which excludes, let's say, disease, or what else should there be?
Great point. So I'm Gary. I wanted to, I like the view of the genome as kind of inspiring, and protein domains were an important functional unit that was identified by comparing sequences and identifying repeated patterns. And then eventually there was a definition of what a protein domain is. It's a evolutionary, functional, and structural unit. And the functional part is kind of maybe parsed out at the gene level for the most part in gene ontology or something with 3 aspects, and maybe you can consider additional ones. But the fact that with genes, at least, there was 3 major aspects of function where it's located, its molecular enzymatic function might be, and it's what process it's part of, is kind of interesting. I wonder, what features of cells, what kind of aspects of cells we should also think about. And, there's a lot of different ones that Sarah, you mentioned a bunch, but some other ones that, we could mention, throw out there are the history of the cells, the future of the cells. So the same cells, same pool of cells could branch off and go in 2 different directions or something. Where, when did they decide to do that? And is that a functional aspect that we need to ascribe to half the pool or something like that? Yeah, so I wonder if there's ideas about what aspects of cell function we could kind of catalog that could go into a future cell ontology with different aspects of function.
So just to clearly follow up to that, but I'll just say clearly one thing people are very excited about is lineage. So I think lineage is something that you almost get people saying RNA is all, you know, downstream of lineage in some sense. And so lineage is the real, I've certainly know, you know, neuroscientists who think that way about cell biology, that lineage is only the real foundational thing because it measures actual causality. And then I would always push back that lineage is downstream of evolution, but I acknowledge that lineage seems to be also technologically what's like right on, you know, everyone's minds in terms of developing new methods. So there's someone else, Mike.
So the problem with function is that it is not kind of cell autonomous in a way in most cases. We need to think of function at a hierarchical scale and always assign, and I would say not only at a hierarchical scale, but also at a quantitative scale. And that's quite different because the genome is an information medium, whereas the body is a physical execution medium of sorts. So if you define function as a phenotypic effect when you remove it, then you do quite well with genes. If you knock out that gene and suddenly the flies do not develop functioning eyes anymore, then you have function. With cells, that is not so straightforward because if you remove a single cell, you will probably never have an effect except if you remove the zygote or the tumor initiating cell. If you remove all cells, even for something that is not directly functional, like structural cells of the body, the organ will just fall apart without saying that the kind of the fibroblasts in whatever liver, kidney, whatever, that are just keeping the everything together are not necessarily deeply functional for that particular tissue. So kind of two dimensions here. The first one is often the function is not it's a single cell, but a group of cells that work together to commit to a function. So we need to be able to aggregate function at anatomical levels. And secondly, the function depends, like knockout is not 100% thing in many cases, but function will always have some dose response aspect to it. And this dose response aspect, as we know from pharmacology, is a very powerful concept. So maybe the thing that is fundamentally wrong with the question of defining cell function is cell as the only unit and function as a binary switch. And we need to kind of cell and anything in between cell and body, and function as something that is in many dimensional and gradual in many ways.
Yeah, and like one of the big problems we have in immunology is often you'll knock out a gene of interest in your population, but there's a lot of redundancy between different immune populations. They can both contribute to certain kinds of host defense. So if you don't see an effect when you knock it out in one, that doesn't mean it's not doing it. It just doesn't mean it has a biological significant change, right? Yeah. Michael.
Hi, Michael Hoffman, University of Toronto. So I'm a author of the 2012 ENCODE paper, so I could talk about it all day. But I will try to stay brief and make a couple words. So I think, you know, a key question, there's not going to be an answer of, you know, what is function that everyone accepts. So you just, you know, the main thing is you need to be very careful about using an operational definition that you can define if you're talking about function. And if you look at the different categories in the Gillis paper, for example, my own view is that things like, you know, evolutionary evidence of function is kind of a you know, lower bound on which parts of the genome in that case are functional, and biochemical activity, as Gillis would call it, or mere existence, as Doolittle would call it, is kind of a upper bound, right? But the mere existence, it's not just that you see it once, it's things that you see over and over and over again that are reproducible. The other thing I will say is, it's tempting to take on the evolutionary selected effect definition, because it's relatively easy to get an operational definition for that, but it doesn't, in my view, correspond to how the term function is used by scientists in general, right? People talk about, say, functional assays, right? They aren't testing evolution in a dish most of the time when people are doing functional assays. So you have to, contend with the fact that there's not a universal definition, and don't try to make one.
Right. No, the goal is to get people's definitions and hear them. I do think that, I do think that there is something to be said about disentangling evolution as directly measured, which is rarely what's being done, and the idea that we think there's some effect on fitness. So we don't think, the dose response idea that was just mentioned is a good example of, we don't think that one cell has an effect on fitness. We think removing many cells has an effect on fitness. And the degree to which it has an effect on fitness is the degree to which it's dysfunctional or functional. And I guess I would kind of be comfortable with saying that a single cell has no function if it never had an effect on fitness in any context, in any way. And it seems like one of the problems, of course, is that, we don't really have a framework. We can say degree of effect, I think, or even agree that there is degree of effect, not binary, but other than the gene ontology, which people complain about a lot, but everyone uses, it seems like people complain about it a lot. Whenever I use it, and whenever people use it, they say, oh, we don't trust the gene ontology, but we've got these great results. But it is the only way that people really have in a structured way to talk about type of function. And so if there's something better for cell types that people would even posit, that would be great to hear or think about. Gene ontology. Oh, sorry, there's a first speaker. I apologize.
It was a great discussion, the way you framed the questions. So I want to look at it from a downstream application point of view, as someone who would think about using this data for practical applications. So everything in medicine there is an inherent error in every measurement, right? Whatever it is. Even disease classifications, if you look at the first classification of lymphoma, they didn't know there were T and B cells, okay? But there was a classification, people were treated based on that. So I think you raised the two key questions of, cell type and function. There are many ways to approach it, and I think, the discussion about, what constitutes function, etc., is very, very important. But I think for a downstream use, what is probably needed more is some level of certainty, in the sense it may not be accurate, but if, there was the talk about cell ontology in the morning. So if the community can settle on some sort of definition for what a cell type is, granted, it may not be super accurate, but, maybe there is a table with that cell saying probability of this being this is this, and here's the list. But at least if we can label it as something, then you can do something downstream with it, looking at, okay, how many of these cells are there and treated versus untreated, whatever. Right? So there needs to be some certainty which can evolve over time. I think function also, I would approach it the same way. Yes, this does not fit well with real biology. And if you say, okay, if you take a germinal cell and a lymph node, there is dark zone, light zone. There are two different types of cells, but within 6 hours, one can transit to the other and can go back, etc. There are intermediate states. Well, if we get into that level of nitty-gritty, then we cannot actually use it. So I would say, even if it's not accurate, there should be some level of classification system that a version 1 and then evolve.
So I 100% agree, if I'm allowed to as the moderator, that like certainty is more important than, it's better, don't want the perfect to be the enemy of the good or the good enough, right? That it's more important that we be certain and concrete about things and have an iterative element, I think is also key, than that it be an idealized system, for sure.
Yeah, I agree with the earlier comments about needs to be dose dependent, and in a hierarchical way, I agree with Michael's comment on how it needs to be context dependent. I remember the skirmish on the other F word, which is fitness. The evolutionary biologists talk about positive selection or neutral, right? And then cancer genome scientists talk about passenger and driver mutation. Keep wondering about, is this mutation oncogenic or tumor suppressive? It turns out just on a single DNA variation fitness can make that protein less fit, can make the cell fitter, can make the organism less fit, or may have nothing to do with between species comparative genomics. So these are different Darwinian arenas. The cell to DNA to phenotype linkage had a lot of slippage. I feel to claim one arena has anything to do with the other requires extraordinary evidence. So I do have try to contribute something concrete. Just about a cell, if a generic cell, if we can compare ourselves to say what are the top 5 or 6 or 7 things a cell will do, you start to name something like proliferation, grow bigger, or move, or die, or differentiate. Just like people say in ballet, a dancer has 5 or 6 moves, or some people just told me tennis players has 14 moves, just chain them together. Those are the functional primals. If we can gravitate to those, then link gene programs to those primitives, then we have some traction.
I think that's a fantastic thought.
Okay, so a couple of people have touched on ontology definitions here, and also talked about how cells don't act on their own. So this is something that actually the ontology community has thought about quite a lot in terms of coming up with standard ways to annotate things. So a standard, one way of framing function that I think is reasonably straightforward is this idea that it's a capability. So a cell is capable of releasing GABA. It's not doing it all the time, but it's capable of doing it, and it's something that you could assay as well. So most of the signaling pathways we have assays for. So in that sense, it's pretty concrete. We could measure things that show us that a particular process is happening, and if that process is happening in a cell, then we can say that cell's capable of that, has that function, it's capable of performing that process. And then more broadly, obviously, cells don't just do these things on their own, and we would. So in the cell ontology, we would say capable of for something that the cell could do potentially in isolation. So, and then capable of part of to put it in the context of a much broader process. So those are the two kind of links that we would have to the gene ontology. And I should say as well, I think when people talk about the gene ontology and reliability, they're sort of mixing a couple of things. One is there's an ontology, which is a structure, which may or may not have a good reflection of the way biologists think, but and then there's the wider Go annotation. So when lots of people talk about Go being unreliable, they're talking about using particular methods for enrichment based on the annotation corpus, which the annotation corpus is variable in quality to some extent as well. And then the approaches of using enrichment are very atomic. You're saying this single gene has been implicated in this process. That doesn't mean to say that the entire set of genes that are required to carry out, say, a signal transduction pathway are there in a cell. So I think, I think that it's kind of to me, a reasonably productive and actionable way of thinking about function. Is there evidence that, that this cell, okay, it's hard to just to take that straight out of single cell data, but is there evidence that a particular cell type can carry out a particular function? Do we have assays that show that?
Do we have assays that show that in context? The context also in terms of genetics, I think that from one of the talks this morning, there was all that beautiful clonal data in tumors, and there that really shows, you can actually assay context, function genetically without thinking about whether the whole organism survives, and you do that clonally. That's something that come from a background in fly genetics. It's something we kind of take for granted. So again, that gives you, I don't know. So I just wanted to put those out there as ways of framing it. Does that make sense?
Excellent.
I'd like to just take a moment and link those two comments. I really like the idea of finding primitives as sort of primitives in terms of cellular phenotypes or functions, which seems linked to the idea of how we would, most kind of generally describe things in the gene ontology, maybe at the highest level, and could also be linked to evolution a little bit in that we think these would be deeply conserved regulatory programs, which was also sort of a part of the message we heard throughout this meeting is people saying, oh, cell types, I don't believe in them, but there's clearly gene expression differences, and those are some sort of regulatory program. And so no one said that exactly, but people said things that were close to that they thought there were clearly regulatory program differences. And so I wonder, does that feel like something that would be achievable, like in the field as a source of agreement, that like, here are 6 cellular properties that, or 10, or even, I hate to say it 100, but you love 6, that are, any cell type, any cell, you could talk about these properties, its degree of its proliferative potential, its stemness, like stuff like that, and people would agree.
Hello. So what I was about to say is sort of like segueing into that. So I'll say what I was originally going to say and then kind of link to what you're saying. So I think part of the problem with our idea of function is the resolution at which we measure the phenotype. So you're talking about, evolution. But the way I'm thinking about it is, say you give some flies like a gain of function mutation. They see better than the other flies. But if it's dark, then there's no fitness difference. So that's sort of why, I understand that we like this idea of conservation. It's like things are conserved, they must be important, otherwise they'll get weeded out. But at the same time, that requires this sort of understanding of the environment and the way that has to come in. So it's not sort of like, kind of like, intentional to the system itself. It's sort of like, sort of cashing into these other features that are coming in. So that's why I think people make this distinction about proximal and distal functions. When they talk about it in concept. So development is proximal, but evolution is distal. But I think there's a slightly different idea of proximal and distal, which is more simple, which is just spatiotemporal proximity and being distal. So for example, a mutation in a gene, a protein gets made, that's pretty proximal. Function or the ability to see better is more distal from that. So I think when we talk about function, there's a matter of like measurement. Somebody said, if you take out a cell, the person doesn't die, but maybe the local the cell network is where the difference is. So I think what we measure changes, what we mean by function. So having said all of that, I agree with you that we kind of need to come up with like a set of features that we're talking about at the level of cell. So I think if that's the resolution, then we can talk about the proximal and distal in a spatiotemporal context, being like, okay, like proliferation, you mentioned as an example. That's a case where if they stop proliferating, we know that's the phenotype we are measuring. So we're not looking at, oh, do the flies die if we knock this thing out? But we say, do the cells proliferate or not? So I think once we determine the resolution of the measurement, which in this case is cell as like the smallest living thing, I think then we can come up with a list of, somewhat general, but still precise enough phenotypes that we can say, okay, when they change between groups, then we can say there has been function. So I think that's the way I organize these thoughts in my head.
So that kind of transitions well to questions 2 and 3 a little bit there. So what are some exemplary processes, for example, that serve as models for unraveling regulatory mechanisms, for example? How do we validate and explore cell interactions and integrate these into our understanding of complex organisms or complex organ functions, right? So what are people's thoughts on stuff related to how we, when we define function, whatever our definition is, how are we going to move beyond kind of these superficial validations of I see that protein expressed, the cell is there in disease and not in health. How do we move beyond that?
I'll just react to your prompt, but also extending from how you framed the question. To talk about ontologies akin to writing dictionary, the good ones take a few centuries to write. But how do you begin? Is it A to Z, or you begin with the most frequent words? So the cell types and cell states and markers, that whole conversation is about building the catalog of nouns, the entities, the to be's. And then the effort to build the catalog of principal actions are the verbs, to do. And then when you get your noun and verb matrix, you can unfold these regulatory mechanisms, stage-dependent effect, different levels of organization. These are the adjectives and adverbs. At least you have a framework starting from the primitives and unfold outward to all endless varieties of biology. I think that's my way of framing it.
One more.
I mean, to me, I think that's when we need to bring in the most geneticists into picture when we define the function, or when we think that we know the function of a cell, then probably we need to bring in them into the picture. They may be able to address some of this through the genetic approach, maybe organ-specific knockout. Then on a rudimentary level, one can think about the organoid culture models to knock them out and start to study. I don't think the single cell community itself can address this at the functional level. I think we need to bring in the other people at the stage to validate it.
Yeah.
So you don't have to start over. The gene ontology actually has been classifying and identifying biological processes that are part of cells for like 26 years. It's not a fixed entity. One can extend it and submit new ideas. It is a perfectly valid way of starting to define what you mean by function. Now here, it's even bigger than that. Like gene ontology has a term for flight, right? Which is a function of like an anatomical set of structures. But it also has going down to the various details of what anything a cell does. So, you could just start by saying a cell is a bag of processes that it does under some circumstances.
I just, is it on? Just trying to bring the conversation to some practical aspects, which, and I think again from my system, which is the cardiovascular system, whatever the function and whatever we want to validate, I think we have a major problem. And I'm asked almost at every talk that I give now, how are you going to functionally validate this? And my answer is a big hand wave. I think for us in the cardiovascular system, but I think in many other systems, we are missing in vitro models that represent the new cell states, let's call them, that we are identifying. So if we can reproduce those, what kind of function we're validating? And how much efforts do we need to put into creating good models? Because the mouse is the mouse, we said it. The fly, whatever we use, is never going to be perfect. So is developing in vitro models that reproduce as close as possible, at least the cell states that we have defined. Because if we don't have that, I mean, at the moment we're doing it, we get the closest thing possible, which is miles away.
Yeah.
We give a recombinant protein and I'm moving to 0.3, or we do a co-culture, but it's a big approximation. And we usually then get down to one receptor, one gene, not the cell as a whole, like it was mentioned at the very beginning. So that is a question that I throw back to the audience. Do we really need a lot of money and time invested in the organoids, in the IPS to reproduce this
phenotypes, first of all, define them and reproduce them.
Right. I mean, that, so I moderated a session for the HCA, I think a year ago or so, or it must be more, it must be a year and a half. And that was the most controversial question, was whether, and it had a lot of audience discussion, whether, what's the more realistic model? Is it organoids that in some ways are faithful and reproducible, critically reproducible, recapitulations, or is it like mice, where that at least are functioning organisms where you get kind of all that multi-scale simultaneously? And I think we certainly see there's a lot of interest, scientifically in both. People are pursuing both. And I thought one of the questions or comments posed in one of the talks, I don't recall, saying that, it really depends on the purpose. Clearly, when you have an organoid, it's controlled for a lot of things. And if you want that control, you might be happy. And if you don't, if you want to have, multiple scales active at the same time, you're lacking that control. And then you might be happy about that too, if that's important to your study. But I at least concur that like having the right system, like co-culturing assays to study particular cellular properties, once we can accurately or reasonably characterize them, not perfectly, would be really valuable. I'll just throw in at my own pitch about these things, that when people pick a good model system, there's a tendency to overfit. So there's a tendency to favor reproducibility at the cost of biological variability. And you can really see that with mouse models, where they discover later that, yeah, if it only works in one mouse strain, it's unlikely to generalize to humans, that we probably would do better testing 2 mouse strains than the FDA requires that eventually. And so in the same way, it does feel like we're at a moment where people are they're trying to get more and more reproducible, which was one of our words. I think, this is, can we get more reproducible models to get more reproducible outputs? And the concern is that if you make the model too reproducible by restricting variance, you actually make it less robust for generalization to new systems. So I would just say, let a million flowers bloom, but hopefully people can get a lot of models going and test a lot of properties, and you can see what's robust across all that variability. And then hopefully that is also the stuff that generalizes better to real biological systems.
Go ahead with a comment.
Yeah, there's just a comment in the chat from Fadel Alakwa. Things we can do to validate permutations and subsampling, enrichment analysis, data generated with other technologies such as proteomics, experimental validation, functional assessment. Yeah, so all very good points. Lots of things there. There's also, of course, a lot of times if we will find a certain function ascribed to a cell or the tissue, there's a huge body of literature that shows, hey, this has already taken place, right? So it's not like it's always starting over, but it's those novel ones, the ones that haven't been there, that I think those are all, is it kind of like lines of evidence in a court case where, okay, well, we see it in different samples. Okay, we can knock it out in a mouse and we see this. Oh, is that enough? Is it enough just to present, here's 4 different ways that are supportive? Because that seems to be the way most of the time we have to go about it.
Just to prompt, not to say to cut off this line, but I'll just say we did have 3 pages of these questions. So I'm just going to turn to the next page. Just not that you have to stop talking about this topic, but just to prompt some discussion on, they're all related. I'm so, not to cut anyone off.
So a couple more thoughts. So when we think about cell function, we should also really use the context of that cell, right? In that sense, I think spatial helps a lot. So I'm curious, so for example, if you are trying to, going back to that cell type and function, which is still, and I like that idea of gene ontology and considering a set of gene ontologies as an ID for a cell. So let's say you're trying to look at the liver, and over decades, anatomists and histologists have described in painstaking detail with electron microscopic evidence of every cell type, right? So we are trying to classify them based on existing knowledge. So there is a structure, there is a location of the cell, there is a certain cell types already defined, and we are trying to fit that. So and then, function is a little more challenging because, that same cell in that location might have, many different functional states. So I'm curious, for those people who have, done the cell typing for the HCA Atlas, what kind of challenges given all this data? Now you have a single cell seq, you know the previous cell types. What are the types of challenges in cell type?
So I'll just start one little thought of mine on that. The one struggle I often have is moving beyond, we're getting gene expression profiles. And a lot of immune cells' final phenotypes are regulated at the protein level. So I work on NK cells and IOCs. At the transcript level, we'll see CD3 being expressed by them, but it's not at the protein level, for example. And these are fully defined by immunologists as distinct populations. One's part of the adaptive arm, one's part of the innate, right? Then you can go in T cells, and beyond it being a T cell, for sure you get expression of a TCR. Then we have Th1, Th2, Th17, all regulated at the protein level by what their phenotype is. So as an immunologist, we always say a defined phenotype is one where you have a conserved transcription factor that when it's there and overexpressed consistently, that defines a new immune cell lineage, right? But if you look at a lot of these transcription factors for T cells, we'll see EOMAs and TBET being expressed at certain points in a Th17 cell, just not high and constitutive, right? So, I mean, I always bump into this problem of I need to put a label on it because I need to put some definition there. It's as good as I can get with the information I have, but is that the way we should be going about it? I don't have an answer to you, but like for me, it's protein. I have to go to protein. Yeah.
Immune system seems the most challenging, but what about the other organs?
So I do a fair amount of work in the brain with the Brain Initiative Cell Atlas. And so we've looked at that data a lot over the last several years. And the brain, in some ways, is a really easy system because, I mean, in some ways it's hard because it's complicated, but in some ways it's easy because you get less cell cycle, right? At least the neurons, not. And, there's exquisite phenotyping and all these different properties, like spatial alone is enough to really say a lot about cell type and spatial in a way that is sort of more richly specific than in other systems, and maybe connectivity and electrophysiological properties, all of which are kind of measurable to some degree. Reproducibility is still a challenge. I think one of the things that was hinted at, again, in one of the talks was that, currently people are stuck on hierarchies. And they like hierarchies because of something else we've discussed, because they kind of look like lineages, which is kind of miraculous in the brain. You're getting mature neurons. You cluster them, they cluster into something that kind of looks like the cell lineage. That's somewhat interesting. And so people like hierarchies, but if you pick different subsets of genes, you get different hierarchies. So you do not get the same, the feature selection is actually rather key. And currently the feature selection is completely, highly variable. We hope for the best. I'll throw one thing out there. You heard the brain has 5,000 cell types. You heard in other tissues, other organs have 100 cell types. And here's a possibility for you. Let's say the brain is actually not one organ. It's 100 organs, each of which has kind of the same cell types in it. That's kind of what it looks like. So not exactly, but you see epithelials you see epithelials all over the place. Those epithelials in different organs and tissues don't have the exact same transcriptomic signature. It's cross-cutting with where it is and what it's doing. In the brain, you see a lot of the same cell types in different locations at what they call the subclass level. And then they're slightly tweaked, and you call them different cell types because they're tweaked. But if they're spatially restricted, as many of them are, not all of them, but many, is that correct? Well, if you did a different feature selection in the gene space, you might say, oh, these are actually the same type because transcriptomically at that subclass level, they're the same. So I feel like this is not even getting to the more interesting problems of cross-modality or deeper thinking about it. I'm just saying, just dealing with the transcriptomic data as is, there's room to really think about, how are we calling the types? Is calling them based on their sort of unbiased, was the big word for a long time, transcriptomic signature. Is that the right approach? It might be. So this all might be the right way to go, and it's good enough, but there's still room to, I think, consider it from first principles.
Stephanie from the University of Toronto. I actually want to come back to the question about the models. And what we do a lot is, I'm actually looking at, so I work in the cardiovascular field as well. I would argue that this is hard to validate in the cardiovascular field because we don't have organoids we can form us from adult stem cells. Obviously, that doesn't work in the heart. And then we also don't do this from embryonic stem cells very well yet. Nobody forms a 4-chambered heart. So if you want to look at congenital heart diseases and confirm anything, we don't have a model. So I would actually strongly argue in that field, we should develop new models. And there's a lot going on. You've heard a bit about the embryo models, and there's people who want to take this further. So I think some of the stuff we can only validate this way, mostly also because when in my lab, we work on the conduction systems of what regulates heartbeat. And I'm actually interested in looking what's different between mouse and humans. We find a lot of stuff is new in our single cell seq that seems human specific. So I can't go back to a mouse, knock out the gene and ask for function. So that's why I think actually human models are super relevant. And in some areas we are better, like in the gut and in the lung or in the brain, we can have organoids, but in the heart we don't. So I think new models are important, not in every field, but in cardiovascular for sure, we've got to build more models.
Let's do one question from the Zoom audience and then one last question here, and then we'll probably have to close. So a question from Sri Devi related to integrating diverse processes. What about morphology and input and output connectivity as key descriptors of neuronal function? Can gene expression inform specific morphological features?
So people definitely are trying to do that. You can capture them simultaneously and build up models that try to incorporate both. The reason people liked RNA, so despite the vast amount of research in the brain and the deep wisdom people have and the exquisite love, I've never heard anyone more in love than a neuroscientist describing their favorite neuron. It is like they are describing their beloved, you know. Despite all of that, it's really very, very hard to end up with coherent categorization outside of RNA. And that's why RNA in the brain became this wonderful Rosetta Stone, right? It's like amazing. Suddenly we have concrete definitions we can all agree on. And despite all that specificity and detail and work, I feel like these other features ultimately people weren't able to fully agree on coherent classification schemes. And so like the Rosetta Stone, the goal is that, yeah, they can be linked through RNA. So that's often why people, if you're doing this, connectivity, sequencing, or spatial RNA, methods or ATAC-seq, like why do people want multi-omes so badly? Is because the ATAC-seq is kind of hard to interpret without the RNA-seq, or harder. And so RNA has become a very useful general modality. I don't know if everyone would regard that as obvious from first principles, but I don't think it necessarily is. It just happens to be true that it's really, really useful to ground other data in that modality. Any last maybe question or comment from the back?
There's one over
sorry,
here.
I apologize.
No, it's okay. I'm Maria from Gary Bader's lab. I was actually more thinking about in terms of integrating. There's so many different definitions and people are looking at function from different level, but maybe as a group, we can come up with like a table that people can start looking at. So if you're more keen to look at protein, then you can define your function based on your protein level or gene or cell. And then everybody just uses these eventually evolving tables. But then you can actually like put a point where you think your function is occurring, because I think it's going to be really hard to figure out what everyone defines function. And it's not fair to say something is not important, even like to the first comment, taking away one cell, whether that's, enough or not. So I just feel like, if you make a table that everybody agrees on and start from protein to gene to cell to tissue and something, and then have it, don't go too far down, but more like location or like some quantity, and then everybody has to fill it no matter what.
I think that's the dream of the controlled vocabulary that our ontology colleagues, so I think there's a lot of consensus there that some degree of controlled vocabulary, however organized, would be useful. So time for no more comments. So we had a third page, which I'll just show, which is it was more technologically oriented anyway. So Sarah, you want to sum up things, or also?
No, just obviously these are ongoing discussions. What's clear from this is that we all don't really have one clear consensus answer on this. But I think it's important to have these discussions, right? To think about it more and more, and to get outside of our own kind of thinking. If we're looking at a disease and trying to identify stuff, well, it's good to take a step back and really think about this from an evolutionary perspective. So just want to thank you all for your fantastic contributions and for being such active participants. And I guess we're ending on exact perfect time. So
enjoy your snacks.
Enjoy your snacks. Yeah.
That's nice.